Good afternoon to everybody. This is the third of the webinars organized within the project E.W.A.S, Environmental and Water Solutions. We have now, today is the 28th of November, and we are hosting as a speaker Dr. Bruno Scanu, who is a forest pathologist, and he will today present a webinar entitled Problems Related on Phytopathological Problems in the Nursery Activities, with particular emphasis on forest tree nurseries activities. So I will just leave the floor to Dr. Bruno Scanu, and if you have any questions, remember you will have the possibility to pose questions at the end of the webinar. Please go ahead, Bruno, is yours.
Hi, good afternoon. Thank you, Professor Quirico Micheli, for inviting me to give this seminar or webinar, how we want to call it. And I'm going to share my screen first. Let me see. This one. Okay. Okay, can you see my screen and my presentation?
We see your full screen, yes.
Okay, so
but you should choose the full screen.
It's not
option.
full?
No. Actually, you are sharing the screen of your computer, so we were.
Okay, so I don't know how to change.
You should just share the presentation. Now we see the full screen.
Not the presentation.
No.
I don't know how to change. Now
you have to go back.
Use no.
Okay, now it's okay. Now it's fine.
Okay, now you see the presentation.
Yes.
Maybe it's not good for me because anyway, need to reduce the. Okay, I try to go ahead like this. Okay, sorry for that. And so my seminar will be about plant health risk in nursery trade. And I will particularly talk about the case of Phytophthora species, a group of pathogens that are really important in nursery environment. Okay, it's not going. Okay, so first of all, I would like to give you a brief introduction of this group of pathogen, the genus Phytophthora. And when we say Phytophthora, we always say fungi, but they are actually not fungi. They belong to a new kingdom, which is called Straminipila, Heteroconthophyta or Chromista, depending on the systematic. They are actually oomycetes, and they are closely related to diatomy and brown algae. So they are closer to plants than fungi. This is an important group of pathogens. As I said, the first species described was Phytophthora infestans in 1876. And it was the cause of the potato blight famine, which caused over 1 million of people died and emigrated because of this epidemic particularly in Ireland and Central Europe. And that is because potato was the main food source of rural communities at that time. Here you can see in this graph is reported the number of species described over time. the first species described was Phytophthora infestans in 1876. And since then, the number of species grow very slowly. Until the end of the 19th centuries, we knew about 50 Phytophthora species that were described mainly based on morphological analysis. And then we went into the era of global Phytophthora surveys, and in particular the molecular, the use of molecular tools to describe the species, and the number of species increased exponentially. Actually, this was also due to the globalization and especially nursery trade that we will see as favorite the movement of these Phytophthora species between continents. And now we know about more than 260 species are described in this genus, and probably there are about 400, 600 species predicted to be described in the future. And this is an important point. I will see the role in particular of the nurseries in spreading Phytophthora species. So it is, when we study a pathogen, is very important to know the biology. And here you can see some of the structures of these pathogens. They produce They produce resting spores, such as chlamydiospores, and they are actually asexual spores. And also they produce sexual spores, which are called oospores. As you can see here, this is the ogonium, and inside they produce these oospores that are characterized by having this very thick wall, which provide them, because of this very thick wall, they can survive for a very long time in the soil. While these are actually the asexual spores, which are called sporangia, and they are they have actually different shapes. They can be papillate, semi-papillate, or non-papillate, and they differentiate within the sporangia the zoospores that are released in the water. And these zoospores are motile because they have these two flagella that give them the opportunity to move into the water. They actually also produce pseudoconidia, we call them, which are typical of peronosporaceae or also fungi in general. And they actually are detached. They are released in the environment, and they germinate directly instead of releasing the zoospores. And here is just the infestation models of Phytophthora species. Most of them are actually soil-borne pathogens, which means that they infected primarily the roots of the plants. And this is a typical features of species that have persistent sporangia. While other species, actually the more evolved phylogenetically, can infect also aerial part of the plants. And this is mainly due to the fact that these species produce sporangia that are caducous, because they produce this pedicel that give them the opportunity to break up from the hypha, from the sporangia spore. And then we have also in between some species that can behave both as soil-borne pathogens or occasionally as aerial pathogens, infecting, for example, the stem and the woody tissues. And this is an animation. I hope that you can see the video provided by a colleague, Thomas Jung. This is a beech tree, Fagus sylvatica. Once the rain starts, the water percolate in the soil. The sporangia release the zoospores, and the zoospores are chemiotatically attracted by root exudate. They incise on the root surface. They produce this coralloid mycelium inside the infected tissues. And then on the root surface, they produce secondary sporangia that release other spores in the soil water. And this is why these Phytophthora diseases are called also multi-cycle diseases. And this is a video under the microscope. This is a sporangium that is releasing the zoospores in the water. They usually produce between 30 to 50 zoospores, and then these zoospores are released in the water, and they move in the water looking for roots to be infected. They can stay alive for about 5 to 7 days, even without host. And this is another video, again, under the microscope. It's quite heavy, so it may take some time to start. And these are the zoospores in a different magnification. And you can see they are moving in the water. And once we introduce in the zoospore suspension in the water a root, you can see that these zoospores are attracted by the roots, and then they start to incise, to attach in the root surface and start to the infection process. And there is also another video, and this has the again a zoospore concentration in the water, and on the top you see two syringes. On the left, one is releasing sterile water, while on the right is releasing root exudates. And you can see that the zoospores tend to concentrate on the right side because of the root exudates. Now, well, I prepared this seminar to be very practical. And after this introduction, I show you first of all, how we can isolate and identify this Phytophthora species, which is not very easy. And then I will talk about also the disease they cause in forest, particularly, and also nursery environment. And I will show you also which can be the solution to reduce the impact of this pathogen. So first of all, you can see here some of the pictures of a training course that we made last year in Tunisia. We collect some soil sample from oak trees. Then we went to the lab and we put these soil samples in plastic boxes, and we cover the soil with sterile water. Then we clean the water surface, and we collect many fresh leaves of different hosts, like carob leaves, citrus, and so on. They have to be very fresh. And then we place these leaves, as you can see here, on the water surface, and we leave them for 5 to 7 days. And after 5 days, you see some of the leaves. Of course, when the soil samples are positive to Phytophthora, they start to produce these necrotic lesions on the leaves. You can see here better. And if you look under the microscope to these leaves, you should see the sporangia produced by Phytophthora. You can use also fruits like apple, for example. You can put inside the apple soil or root samples, and afterwards, after a few days, you start to see these necrotic lesions on the fruits, and you can isolate them using selective medium. You can do also a direct isolation from necrotic lesions on the roots or other plant tissues. So once you get this necrosis on the leaves or on the plant tissues, then you have to put them or place them on a selective medium specific for Phytophthora. So we prepare usually this medium with different antibiotics that avoid the growth of, for example, bacteria, fungi, or other oomycetes. We usually look, once we put the small pieces of leaves or plant tissues on the selective medium, we then check after 24 hours whether there are any hypha growing, and we do this under the microscope. And if we see typical Phytophthora mycelium, which is very typical because it's coralloid, as you can see here, then we subculture in PDA or carrot agar, for example, and then we wait until the colony grow. Once we morphologically, if the colony look like Phytophthora, then we start with the identification that can be based on morphological analysis. So we look at the structure that I showed you before, like sporangia, chlamydiospore, alpha swellings, or ogonia, or as now very often we do, is the molecular identification. So we extracted the DNA and we sequence some specific gene regions, and then we analyze the sequence that we obtain. And we compare them with those deposited in international database. So just a few examples of Phytophthora diseases in forest. One of the most notorious example is Phytophthora ramorum, which is a quarantine pathogen. And here you can see a disease that ramorum causes, which is called sudden oak death of a specific oak species in California. This pathogen was basically most likely introduced with these plants that you can see here, which is an ornamental plant, rhododendron, introduced from Asia. And this plant species act as a secondary host because these pathogens infected the leaves, but it doesn't cause any real disease on this host, but produce a huge amount of inoculum, of spores. And these spores, then they jump to another host, which is oak, where it causes these bleeding cankers on the stems and the death of the trees. A similar disease, which is called a sudden large death in the UK, also caused by Phytophthora ramorum. Very similar symptoms, aerial infection, but in this case, the host, Japanese larch, which is an exotic species that is cultivated in the UK, is acting also as the host where the pathogen sporulate. And this is another interesting and important disease. It's a cowrie dieback, caused by Phytophthora agatidicida in New Zealand. This species, cowrie, is endemic to New Zealand. It's a conifer tree. And there in New Zealand, there are many very old trees. And actually, this species is going to disappear, unfortunately, because of these pathogens that you can see cause these resinose cankers on the stems, and then the tree died. Another example is Phytophthora cinnamomi in Australia, where this species is considered a key threatening process to the Australian biodiversity. That's why, because this species in particular has a very wide host range, so can infect many species. So once it was introduced in this natural ecosystem in Western Australia, there was a huge impact on the ecosystem, because many species were susceptible to this pathogen. So there is really an impact on the biodiversity. In fact, some of the plant species and also fauna are at risk of extinction because of the impact of these pathogens. Just to give an example, this particularly pathogens in Australia is called a biological bulldozer, because the destruction that causes in the native ecosystems. This species arrived also in Europe a while ago, was introduced the first time in Portugal, and spread very quickly across Europe, infecting in particularly chestnut trees, which is an important forest species in Europe. And here you can see the main symptoms. Phytophthora cinnamomi cause the so-called ink disease, because of the these exudates that are produced around the collar and the roots are typical of the disease. And also these flame-shaped lesions that we often see in diseased trees. And this is the results of the disease. Many chestnut trees can die in a very short time. Phytophthora cinnamomi, once was introduced in Europe, it spread also on other Fagaceae species, in particularly oaks and Quercus suber and Quercus ilex, causing very huge damage, particularly in Spain and Portugal in the beginning, but now it's spreading in other countries in Europe. These are the symptoms, the main symptoms on the crown, on the top part of the trees, which are often associated with, of course, root symptoms, where actually the infections are. And this, in these pictures, you can see the lack of fine and lateral roots, also necrosis, and sometimes canker, necrotic lesions. And this infection occasionally can spread up to the collar in the stem. And you can see here a young cork oak trees showing these bleeding cankers. This pathogen in particularly in Sardinia, where we have many large areas of cork oak because of the cork production. This pathogen is spreading in the new plantations, and this we'll see it's strongly related to the nursery, the use of infected plants. This is, for example, is a large plantation, more than 500 hectares of cork oak that was planted 20 years ago within some European countries, programs, and you can see the high rate of mortality due to the infection of Phytophthora cinnamomi. We did also some molecular study, population studies on these particularly pathogens, and what we found is that if you look at the genetic variability of population, East Asian population, like in Taiwan and Vietnam, we found a huge genetic variability, while the populations that we have in Europe and in the US, they are basically clonal. So that means that this species was introduced from Asia Europe and US. And this is actually the case of many other Phytophthora species, like, for example, Phytophthora ramorum. And we prove that these important pathogens also originated from East Asia. You can see here some pictures of Vietnam and Taiwan, where we went a few years ago, very remote zones, areas, and you can easily isolate the Phytophthora ramorum from soil sample collected around healthy trees. So the pathogen is there, the species, but it's not causing diseases. That is mainly because the species have co-evolved with the host. While once it is introduced in a new environment like Europe or US, the host, the plants species are not co-evolved, so they very often are susceptible to the pathogen. And I showed you before the number of species that have exponentially increased over time. And here you can see the number of disease caused by these pathogens that you can see also have increased in the over the last years. And there are many studies that show that there is a strong correlation with the increase of disease with the increase of living plants that are moved across continents. So this is probably one of the reasons why also these species are increasing in our environment. If you compare these two graphs, you see how the living plants, the movement of living plants between continents have increased over the last years, and also the number of oomycetes and also other fungal species have increased over the last years. So once the movement of plants between continents usually involve the nursery that receive and then spread the plants in the areas. And here you can see some of the data of the number of movement of plants between the continents. And you see in this graph, for example, that every year we import plants from Africa, for example. Over 1 billion of plants arrive from Africa to Europe, or from Asia, Russia, North America, and so on. And most of them are ornamental plants. So there is a big business in plant market in the world. And once a plant arrives to Europe, because of our system, the plants can easily move across the different countries. So you can see here the network with the Netherlands, for example, the main country receiving plants and then spreading in other European countries. And a few years ago, we made a study with many colleagues in Europe, and we demonstrate the widespread Phytophthora infestation in European nurseries that put forest, semi-natural, and horticultural ecosystem at high risk of Phytophthora diseases. Here are some of the data that we obtained. I'm not going in detail to that, but this graph shows you the number and the diversity of Phytophthora species that we detected in the nursery. And those species that are marked in red, they are actually considered exotic species. So in nursery, actually, we really see how these exotic pathogens are widespread. Of course, once we have an infected plants plant in the nursery, and then we sell out, we bring out from the nursery, and we plant into the forest, we got these results. For example, these are oak trees in oak plantations, or also, for example, if we use If we use the ornamental plants, this is a garden in Sardinia, and these are infected. This is the results. We also made a study in collaboration with a colleague in California about the restoration sites, for example, area that are damaged by wildfire. Of course, there is an intention to recover this area by planting, but if you use infected plants, then there is a risk of spread new disease, and this can be even more challenging than a fire. So going specific to the nursery, what we can find in the nursery here, I will show you first of all some of the Phytophthora disease symptoms that we can see in nursery. Here, for example, you can see seedlings of Pinus pinea, which is a common species in our forest, and it is one of the most conifer species used in the urban streets and areas. And you can see here a huge damage caused by Phytophthora cryptogea on Pinus pinea seedlings. You see here, and on the right side, you see particular of the roots infected by Phytophthora. You can see the necrosis on the main roots. Also oaks, we grow many cork oak and olm oak seedlings because they are the main oak species in our environment. And you can see here some seedlings already dead and others showing chlorosis or yellowish leaves, which are the early symptoms of infection. And if you look to these seedlings, if you look at the root system, you see the damage, the necrosis, and the lack of lateral roots. This is another species, Myrtus communis, which is very typical of our Mediterranean maquis vegetation. It's probably the only Myrtaceae species that we have in Europe. And you see, this is a very susceptible host to Phytophthora, to different Phytophthora species infections. And you see the symptoms here: chlorosis, dieback, and the death of the plant. Also, another example, Viburnum tinus this is an ornamental plant that can be easily infected by different Phytophthora species. Also, aerial infection, as you can see here. This is, for example, Phytophthora ramorum infections on the leaves. So, why Phytophthora is so widespread in nurseries? There are many reasons for that. First of all, because of the environmental conditions. In nursery, you usually have the plants are irrigated, so you have water in the soil, and that's what Phytophthora need. Usually, temperature is mitigated in winter and in summer, so that's favor the infection of Phytophthora species. And then in nursery, in a very small space, you have many plant species. So there are many opportunities for Phytophthora to infect different hosts. But now I'm going to show you which are the main hazards for Phytophthora contamination. And first of all, I have to say that very often we see that plants, the seedlings, are grown on the ground, and this is a high risk of contamination because of, as I told you, Phytophthora is in the soil, is a soil-borne pathogen. So if you grow the plants, the seedlings already in the ground, it's easier to get contamination if Phytophthora is there. And these are just a few examples: chestnut, walnut, and cork oak. Another critical issue is the potting media that we use in the pots for the seedlings, for the plants. And this is also considered a high risk of contamination. And you see here, usually in nursery, they bring the soil, the potting media from outside the nursery, and we need to make sure this is free of Phytophthora. So we can make some analysis once we collect this soil. But then the soil has to be stored somewhere in the nursery. Usually it is allocated outside in some area, and we also need to make sure that, for example, if our soil is free of Phytophthora, we have to store it in areas where Phytophthora is not present. And even if we allocate the soil inside, and you see some of the sample, maybe the soil that we bring from outside the nursery, and we put it close to other substrate, we have to be sure that these substrates are free of Phytophthora. Otherwise, it's very easy to get the soil contaminated. Another critical issue is the standing water and flooding. This is also a high risk of contamination, and that's, as I show you in the beginner, is the water is actually needed to close the life cycle of Phytophthora. And if you have a situation like this, these are perfect conditions for sporulation, for Phytophthora spreading. Or here you can see, and I put some species that we got from this water. So even a situation like this are not are very suitable for Phytophthora infection. Another critical issue, which I call possible contamination, is the irrigation water. And very often in nurseries, the water comes from lakes or rivers. And when we use this water, we need to make sure that also the water that we use for irrigation is free of Phytophthora. Another hazard, very important hazard, is the cool pile and green waste that we normally have in nursery, especially those seedlings plants that died because of disease or other causes. They are often accumulated in some areas, and in worst case, they are even recycled. So in nursery, they reuse this organic material, and this is a big mistake, because if you have infected soil and you reuse that, then you get contaminated the new seedlings. So these areas have to be treated careful. Otherwise, you further spread the inoculum, as you can see here in this picture. And then I move to the possible solutions to this problem in nursery. And we call these best biosecurity management strategies. I told you that one of the main hazards is the ground contamination. So you need, when you make a new nursery, or you want to change how to grow your plants, you need to make sure that the soil is free of Phytophthora. So one thing that we can do is actually use a gravel layer, especially to avoid the standing water. Or you can use also some material that can avoid the flooding water, but also isolates in some way the plants from the soil. And here is what we made in a specific project that we have since 2 years with local forestry agencies. And we use this, we actually raise the containers above the ground, to prevent the standing water and the soil contact. So we pull up the seedlings in this bench of about 1 meter or a bit less, depending on the situation that we had, and we grow the seedlings on this bench without contact with the soil. We also use new containers when possible, or properly disinfected used containers, and we did that for reducing the cost and reuse the pots. For sterilization of the planting media, the soil, or also the pots, we use steam sterilization, or we can also use sodium chloride at 5% concentration, which is quite good to sterilize, for example, the pots and the tools that we use. Another hazards we saw can be the irrigation water. And what we did in our project is to use, possibly when you can, a deep well water when the risk of Phytophthora contamination is very low or null, I would say, or adopt a filtering system for irrigation water if you are using water from rivers or lakes. Or also very important is to store the irrigation water in disinfected tanks. This is also very important, and we clean, in this case, the tanks. So we were using basically well water when we can, store in these tanks that were disinfected before adding the water. We can also recapture the runoff water to reuse, of course, the water and treat it with approved methods of sterilization. Other preventive measure for contamination. This is also very important because we need to sterilize the soil, we need to filter the irrigation water, but then we have to use some preventive measures to avoid contamination that can happen even later in the chain production. For example, cultivate plants from seeds or import seedlings only from nurseries that are applying the same growing conditions. We need to implement strict hygiene measures, and also like cleaning the boots, the shoes of the people that are working in the nurseries, especially if they are moving from a forest to a nursery, or also sterilize, disinfect the tools that we use in the production. And also very important is the phytosanitary, to have a phytosanitary monitoring plan, and you can use rapid diagnostic kits if you have symptomatic plants, and also very useful to record all the plants that you maybe see with Phytophthora symptoms. Another important task that we found is very useful to produce plants free of Phytophthora is to put effort into nursery staff training and keep them updated. It's not an easy task, but it's very productive. Also, staff knowledge of plants and their needs is the key to ensure quality production. Especially in this picture on the right, you see a staff, a technician of the nursery that was explaining to his colleagues how to produce plants free of Phytophthora. And yes, we did many courses, practical courses, moving into the nursery and showing Phytophthora symptoms or other diseases as well. We took also sample, and we made some open labs in the nursery, so showing how we can isolate Phytophthora, and also very important how we can recognize when Phytophthora is present. And these are the results of this project. And you can see the plants, very healthy plants, I would say, on the benches, produce in this nursery that now are using those guidelines, best biosecurity practices that we developed and implemented in our projects. And maybe I finished with this picture. This is a chestnut I showed you before. That is a very important forest species in Europe. And for example, in Sardinia, forest nurseries were unable to grow this species because of inky disease. And with our project, we showed that actually chestnut can be grown. And you can see here the seedlings, 6 months seedlings, very healthy, very green. And if you look also at the roots, they are very, very healthy. This was very good outcome of our project. Yeah, so just to underline why is so important for us, but I think also for you and other countries to get healthy plants, to produce healthy plants. For example, to face with the climatic crisis, there are some a new European forest strategies for 2030, which is to improve the quantity and quality of European forest by planting 3 billion trees across European countries. But this is, of course, is very useful, a very good opportunity, but we need to plant healthy plants. Otherwise, we are just spreading disease and causing more damage than benefits to the society. I think I very late because it's already 1 hour. I had some other slides about a life project that we have, but I don't know if I can talk for about 5 minutes more.
Bruno, of course, yes, you can take 10 minutes more, and then we will keep 5 minutes for questions.
Okay, I will go very quickly through these slides, which is about a life project that we started because, of course, it's so important when we plant forests that we use healthy plant material. But very often we already have the disease in the forest. So we need to do something. And nowadays, it's very difficult to treat the forest. And in this project, we are developing some nature-based solutions, which are based mainly on the use of microorganisms or biostimulants or biofumigants to control Phytophthora diseases in forest. And particularly in this project, we tested them in mesocosm trials in the beginner, and then we move into the forest. These are the products that we use. They are mainly products already available in the markets because they have to be available also for the stakeholders, and they are Trichoderma species, for example, or Bacillus species, but there are also some biostimulants like phosphonates or a biofumigant like this biofence that we use in the project. Well, we test first in the mesocosm, and then we move into the field. So there are some experimental plots across 3 different countries: Italy, Spain, and Portugal. There, where we are testing this product, of course, where Phytophthora is present. These are some photos of the treatments with a tractor, with a drone, where we cannot use machines, or you see also spreading the crown, or spraying the crown, or the soil and the different products also. We are also applying some preventing measure, which means to prevent the introduction of Phytophthora. And as I told you before, they are mainly soil-borne pathogens, so they live in the soil, and we need to avoid that infected soil maybe arrive into a free area. And what we use is this kind of carpets, for example, that you see here. So all the machines that go into a forest that is free of Phytophthora, they have to clean the wheels, like, for example, tractors or cars. This is very useful in some areas like national parks that are in the project. This, for example, is one of the biggest cork oak forests in Europe, which is called La Almoraima in southern Spain, in Andalusia. And you see this cleaning station for the machines. We also developed this cleaning station for boots, for hikers, for example, people that go into the parks for walking. They have to clean their boots to avoid the introduction of Phytophthora. These are some of the examples in Spain, Italy. And very important, what I show you also for the nursery projects, to communicate, to disseminate, to inform the people, to arise the awareness on the risk of spreading these pathogens and new disease into natural environments. And we did a lot on this particular project, a live project, which is called Phagesos. And we did so many training to stakeholders, schools, open days, meeting, conferences, and so on, to spread our scientific knowledge to the stakeholders. These are some of the events that we usually organize in Sardinia with students. We go very often into the forest that show diseases, so we can show the impact of these pathogens. And of course, within this project, we have to monitor scientifically what the efficacy, for example, of the treatments that we are implementing in the project. And these are some of the activities that we are doing for the monitoring, using, for example, multispectral cameras on drones, or assessing physiological parameters on the treated trees, and so on. So concluding, just a few remarks. Forest nurseries still represent a major pathway of introduction of exotic and invasive Phytophthora species in the Mediterranean countries. The current EU directives on urban forestation and forest restoration underline the importance of producing plants free of pathogens. And also very important, the last few slides that I show you is the importance to develop new technologies for preventing and controlling Phytophthora disease. I would like to thank you for your attention, and also here is the list of institutions and people that are collaborating in this project that I would like also to thank. Thank you.
Thank you so much, Dr. Bruno Scanu. That was really, really interesting. I think that this is also relevant because within this E.W.A.S project, one of the activities is related exactly to the setup of a nursery for forest trees plantation, and the phytopathological aspect, the phytosanitary aspects are really paramount in this case, because as you say, this is useless to plant trees and then to spread the disease through these trees. So the trees that are going to be produced in the nursery need to be absolutely free of pathogens. I would like to thank you, and I would like to thank also the audience. This webinar has been recorded and is being recorded actually. So the recording will be also downloaded in the and webcasted through the platform of NRD. And I will provide you the coordinates to access this webinar. Before we close, I would like to ask whether there are some questions by the audience. I see two people. I don't know whether there are some others on the other side. Do you have any questions?
Yes, hello.
Hello?
Yes, I have one question.
Yes, go ahead. If you can, you should approach the microphone. Otherwise, it will be difficult to understand.
Yes, I was like,
Good, thank you.
I would like to know if those same pathogens also infect the trees which are here in a tropical climate, because what the facilitator was talking about, most of them are in the let me say in the cold areas, because we have different climate zones. I don't know if it is the same case here in Africa, so I would just like to know about that.
Yeah, I hope I.
The question was whether
Yeah,
these kind of pathogens are able to infect the tropical forest trees.
Actually, yeah, I have to say that tropical areas are very interesting to study. As I show you in the presentation, some of the species actually originated from the tropics. Usually in the tropics, you have water and mild temperatures, which are perfect conditions for these pathogens. So I'm aware that even in Africa, in Central Africa, there are problems with Phytophthora. For example, Phytophthora palmivora, it's causing huge damages on cocoa production, cultivation. So definitely there are species, Phytophthora species, that are causing disease there. And especially as Professor Micheli was saying, if you are planning to develop some nursery activities there, it is important, especially if you are starting, maybe to use some of the strategies or measures that I show you before to prevent infection by Phytophthora. And this is also important because if you are cultivating agricultural crops, it's good to have healthy plants because they are more productive and you are not going to get problems, phytosanitary problems then. So I presented mostly diseases in Europe, but I'm sure there are many diseases caused by Phytophthora also in the tropics.
If I may, Bruno, what you said about Phytophthora is perfectly applicable to any other soil-borne pathogen, and that can be propagated, even other pathogens like Rhizoctonia, Sclerotium, Pythium, Fusarium. All of these kind of pathogens are affecting trees and other plants, of course, and they are transmitted by the same way. Maybe they may be less transmitted through water, some of the species, but in most of the cases, the strategies that you design are effective also to prevent other species.
Yeah, I fully agree. I fully agree. I didn't mention maybe in my presentation, but what we notice in the nursery is actually talking about with the people working there, that there was a reduction also of other diseases. So when you use these strategies, you also reduce other possible diseases, not just Phytophthora, which is, I think it's good.
Are there any other questions, my ladies?
Not really.
Okay, so if not, I thank again all participants, and this webinar will be recorded, as I told you, so it will be available. I hope that before the next webinar that will be taking place on the 12th of December, that is the organization of nursery based on this concept. So some of the things that Bruno has already told and explained you will be recapped and put it into practice. And so it would be useful if your colleagues that could not be present today will look at this webinar during the next weeks so that they will be aware of the problematics of the problems in order to understand what are the solutions. Okay, so I please you to tell them to when the webinar will be available online to assist and to open it and to assist to this interesting lecture. Thank you so much.
Thank
you, Bruno, again. And see you on December the 12th for the next webinar. Bye-bye.
Bye-bye. See you.